Thanks so much for the kind introduction and for the opportunity to present my work today. And then as soon as the slides are up, we can get started, because I think I'm the last talk before lunch, so I want to make sure we all get to lunch on time. So I'm going to talk today about our work to develop a streamlined, optimized protocol to do a sort of a variant of saturation prime editing that we call curated loci prime editing, or cliPE. And before I get started, I wanted to shout out a very talented technician in the lab, Krina, who worked very closely with me on this project. And so most of the data I'm presenting today is Krina's. Okay, so let's jump in. I have nothing to disclose. And so by the end of today's talk, my hope is to introduce cliPE, what it is, how does it work, most importantly, because I don't really have the time to get into the super technical weeds in a 12-minute talk, I want to give broad overviews of how does it work, how much does it cost in terms of time and money, and then if you're interested in doing it, just let me know and we can
doing your thing.
Oh, okay. I'm happy to talk more about technical details either at the poster session, I'll have a poster, or you can contact me on email or Twitter. And then I'm going to give an example of cliPE on a single gene, TSC2, where loss of function variants cause tuberous sclerosis. So as I mentioned, this is a variant of SPE, so I want to give a shout out to all the authors of the SPE paper if they're either in the audience or watching online. Very inspirational and aspirational paper for us. Okay, so sort of the upfront questions, why not just do SPE? And the answer is mostly cost. I didn't have much funding for this project, and so I was trying to figure out how could I do something like SPE, but within my budget. So TSC2 is quite a large gene, 42 exons, over 1,800 amino acids. I apologize, the math is a bit off. I didn't catch that till this morning, sorry. But the point still stands that trying to do SPE was going to use up most of my budget and not leave a lot left for salary. And so I started thinking, which is kind of important. It's nice to pay folks for their work. And so I started thinking about what's a reasonable alternative that's a compromise where I could still get data on enough variants to perform VUS resolution, which is the ultimate goal, right? But with using smaller oligo libraries. And so the first analysis we did is basically just to tile across the TSC2 gene and ask, how many variants in these sort of 10 codon regions that you can do prime editing are actually sort of relevant for VUS resolution? So this is inclusive of benign variants like synonymous variants in ClinVar, or sorry, synonymous variants in NOMAD, or known benign or pathogenic variants in ClinVar. And we found that most of these 10 codon stretches rarely exceeded 50 variants. So rather than pay for saturation and get hundreds of variants, we could just make a bunch of small libraries tiled across the gene and still get enough variants to do our project. So that is what we did. Let's see if I can get it to advance. Sorry. Okay, so here's cliPE, the method overview. Hopefully some of the folks of a certain age in this audience will recognize the bottom right. So how this works, we start with a prioritization. We try to get the most bang for our buck. And so we find regions that are roughly 10 codons in length that have the most, are the most dense for VUS. And we also make sure that these regions have some truth set variants so that we can calibrate our assay. And we also put in synonymous variants from NOMAD, premature truncation codons tiled across those 10 codon regions for assay calibration. We also look for guide RNAs that are predicted to have activity so that you're not wasting your time making a library for a guide RNA that won't do anything. We then, even though all of these guide RNAs have predicted activity, we still do an initial screen of all of our guide RNAs that we would use for the prime editing screen. And the reason that you do this is even though these libraries are not that expensive, I'll show the prices in a bit, you still don't want to waste your time and effort on a library that won't actually generate any data at the end. And so I will fully admit this is a tedious part of the protocol, one I would love to get around, but so far there's kind of no way around it. Oops, sorry, let me back up. Let me get through the rest of the protocol. Okay, so once you have screened your guide RNAs and you know that they work, you can then design and clone libraries around them. This is fairly straightforward. I'll go through how much time and money that takes in a minute. Once you have those libraries, you put them into haploid cells. We use hap1s in our lab, but you could make locally haploidized lines like has been done in the SPE paper. You then want to do your selection, just like we would do with any MAVE. We do a fluorescence activated cell sorting assay, but you could do, this should work with theoretically any selection method. Your final sequencing is very cheap in this style. We can just do targeted amplicon sequencing, one sequencing run for your entire experiment, which is great. And then you do a final step where you use jellyfish to count all the k-mers that go along with each of your variant of interest, feed that into the Fowler Labs Enrich2 software to get your final enrichment scores. Okay, so prioritization, very fast, very cheap. Screening spacers. This takes a little bit of time. It's a bit tedious, but it's not that expensive. The cost here that I'm showing is assuming you don't have any of the reagents to do this in your lab. So once you buy into those reagents, future screens would get quite a bit cheaper. The designing and cloning of libraries is mostly waiting to get those libraries from either IDT or Twist. Once you have them in hand, it's a very quick PCR, single-step cloning overnight. You'll have your MIDI prep to put into cells within 2 days. You will also want to QC that, so you might not want to necessarily put into cells on the second day, but you theoretically could. Transfection is really just the cost of your transfection method. So we use the Neon for hap1s. It's about $20 per replicate. It's not too bad. Selection is going to vary wildly depending on what your selection method is. I'll just give our example for our cell sorting. Because these are relatively small libraries, each of your sorts is quick. We never sort for longer than 15 minutes for a single replicate. We always multiplex, do multiple replicates at a time, and so in like a 1 to 2 hour fax session, you can usually get through like 5 to 8 replicates. As I mentioned before, the amplicon sequencing is very simple, cheap, and easy, and you can do it all in one go for less than $2,000. And then the actual like final bioinformatics is fairly low cost, assuming you have access to compute. And it's also very easy to do, which is awesome. Okay, so overall, when you add all of it up, you should be able to do this all in less than 16 weeks for about $1,300 per library, assuming that you do at least 5 libraries. So it's not the most efficient compared to something like SPE, but when your budget's limiting, I think it's a reasonable compromise. So I'm going to briefly go over a quick example with the TSC2 gene. So we initially screened about 12 pegRNAs. We just picked a single premature truncation codon for each of these exons and asked whether we could put those into hex cells efficiently. And then we assumed that if we could make a single variant, we would likely be able to make a library of variants. I won't go through the data down here, but we essentially got about 50% of our designs to work, so we ended up with 6 libraries that we moved forward with. So then we had to figure out a way to select for TSC2, and so for anyone who's familiar with tuberous sclerosis, they'll know that the TSC1, TSC2 protein-protein complex is an important inhibitor of Rheb, which is acts to sort of keep mTOR signaling in check. And so when you have a loss of function variant in TSC2, it leads to constitutive activation of mTOR and phosphorylation of its downstream targets. So we designed a cell sorting assay based around phosphorylated S6. Assuming that our TSC2 loss of function variants, any cells that had those variants would have high levels of phospho S6. I'm going to skip that for time. So here's what the overall results look like for the full experiment. So this is over 200 variants. About half of those are for assay validation calibration. The other half are VUS. So what I'm showing here are sort of Finley lab style plots, as the best we can do at the moment, where we plot the amino acid position on the x-axis. So you can see that these are roughly 10 amino acid stretches. We have the enriched to function score on the y-axis. And then I'm showing dotted lines for either the variants that we have premature truncation codons, or the median across all of the variants in that exon, or the median for all the synonymous variants in that exon. And you can see that for all of the exons except exon 42, we have a really nice separation. Exon 42 is the last exon of TSC2, and the premature truncation codons there are actually predicted to retain function. Okay, and so what if we sort of collapse all the data together instead of looking at it exon by exon? We can make histograms and see that our synonymous variants separate nicely from our premature truncation codon variants, with a little bit of overlap in the middle of the plot. We can then use this data to draw 95% confidence intervals for the distributions, and then assign at least pathogenicity to the rest of our data set, which are our ClinVar truth set or our VUSs, which is the plot on the bottom. So our ClinVar truth set BLBs are in blue. Those are mostly on the left of the plot, which is exactly where the synonymous variants lie. And our known pathogenic or likely pathogenic missense variants are on the right side of the plot to the right of the pathogenic threshold. Our VUS, for the most part, lie on the side of the plot where the BLBs are, suggesting that many of the VUS in TSC2 are in fact likely to be neutral variants. We do have a few that have scores that are suggestive of pathogenicity as well. I think the number's somewhere around 6. I'm going to skip that also for time. So just to summarize, cliPE is a method for subsaturation MAVs. It uses haploid cells and prime editing. It's basically a variant of SPE. So what I think the strengths of cliPE are I think it's good for a lab's first MAVE, which I realize is not going to be the case for a lot of people in this audience, but if you have colleagues that are intimidated by MAVs, maybe this is a good option for them. I think it's good for like generating preliminary data for an R1, where you're then going to do more variants, but you've already shown that your protocol works. It does not require a huge budget. I was able to do it on the $50K budget that I had. The hands-on time is actually much less than the experimenter time. So even though you're going to be working for 16 weeks, you can definitely juggle other things. There's a lot of just kind of waiting for the cells to do their thing, so to speak. And then there's also a lot of pause points where you can stop, go off to a conference, and come back and restart without losing tons of momentum. And I think the main trade-offs relative to something like SPE or SGE are that cliPE is definitely going to be retrospective. It's going to be based on the variants that are present in Nomad and ClinVar at the time that you design your libraries. And we know that those databases are always changing, especially we just had the Nomad version 4 release that changed the game pretty dramatically. And so that is sort of, you need to know that going in when you make that decision, whereas SPE or SGE is going to be both retrospective and prospective. And so as far as like if you want to try this out, or if you have a colleague who might want to try this out, we'll have a preprint up shortly on the TSC2 example. It's going to have what we think are fairly detailed methods that you could reproduce this. We're also thinking about writing this up as a methods article with even more detailed methods step by step to try to make it as easy as possible. If people are interested in it, please let me know, just because I want to make sure like it's worth our time to do that. But I'd be more than happy to do it if people think it would be interesting. And then we'd love to collaborate with groups to kind of make a second generation of cliPE that adds more selection methods that are stuff that our lab currently doesn't do. With that, I'd like to thank my lab. They're a great group of folks to work with in Chicago. My funding from the AES, and I'd be very happy to take questions. Thank you for your time.
All right, thanks, Jeff. Questions? Start with one. So efficiency is never as high as we want it to be when we do prime editing. Is
That's
that
fair.
Yeah, so did I catch that you recommend screening the spacers in HEC 293?
Yeah, so we do like spend 2 weeks pre-screening the spacers to make sure they have activity, because otherwise, yeah, you're gonna waste your time. Yeah, I would say so. For at least for TSC2, we found that we generally get somewhere between 15 and 30% prime editing efficiency, and it's very dependent on which, like, the spacer and the primer binding site and the RTT, all that stuff. But even 15% is more than enough to do the experiment. So.
Maybe have one more. There's a proliferation effect as well as phospho S6, did you say?
That's a
TSC2?
good question.
And do they
It's
correlate?
unclear. So if you look at like some of the genome-wide screens of what is, I'm blanking on the name of the database, but if you just use like siRNA to knock down TSC2 or you knock it out, there does look like a little bit of shift, but it's only in certain cell types. And so, yeah, we didn't feel comfortable enough with like that data being robust to turn this into a growth assay. At least not in HAP1s, maybe in another cell type. But it's a good question. I wish I could have done a growth assay. The FACS is not too bad, but it does end up being technically a little bit challenging.
Thank you for the talk. Super interesting. So it seems like this prioritization step at the sort of very beginning of the pipeline will more or less very much like dictate how much interesting data you can get out from this sort of screen. And so I'm just curious if you have any maybe like tips or frameworks that you had used during that prioritization step that other labs could think about when they are also trying to implement a similar method.
That's a fantastic question. Yeah. So thoughts, I would say we didn't do it in, I think I think there's a cleaner way you could do it. We kind of roughly separated the gene into regions where the local VUS density was high, and so we were looking for a minimum of 40 VUS that we could potentially reclassify from any one design. We didn't always quite hit that mark. I think some of them are more in the 30s. And then we also wanted, in total, our truth set to be at least 25 variants and a split between BLBs and PLPs. And so we tended to not pick designs that didn't overlap any ClinVar truth variants, because that's a bit frustrating. The other factors were like looking for spacers that had high activity in like in silico predictors like CRISPIC. I forget, there was another one that we use as well. And I think our thought is like if we were to write this up as a methods paper, we might actually try to write a little script that could help you like do the prioritization so that it's not quite as ad hoc. So if that's something that anyone's interested in, please let me know, because like I don't think the script would take that long to write. It's just like would people actually use it is the question, right?
All right, so I think we have one more. So thank you, Jeff.
Thanks.